Okay, everyone, thank you and welcome. I'm Jeff Agar, your co-host with Paul Dennis, who's here somewhere. It's my pleasure to introduce Anthony. Many of you guys already know, but he worked with one of the coolest cats in top-down, Lloyd Smith, got his PhD there, and is now a postdoctoral scholar in Emma Lundberg's lab at Stanford. And if you didn't know, Lloyd's at Wisconsin. I'll give you the quick spiel on the Consortium for Top-Down Proteomics. If my slides will advance. If they don't, I might just turn it over to Anthony. There we go. So here's our sponsors, some of them. Thank you all very much. Just a little notice, get ready and we're going to be having the second International Top-Down Proteomics Symposium in the fall, October 3rd and 5th at Northwestern. I hope everybody will be there. And it's looking like a good program so far, and still putting it together, but a lot of cool fun. And plus, you'll be at Neil's place, and he said free pizza for everyone, Chicago style. And here are the ways that we're contributing. Most recently, the Human Proteoform Atlas Initiative and Native Top-Down projects are going. And that's the spiel for the Consortium for Top-Down. I will let you take over, Anthony, and look forward to your talk.
Thanks, Jeff. Yeah, I think can you unshare, and then I'll. Yeah, that's great. It's working on it. There we go. Yeah, it's lovely to be here with you all. It's a great topic for a seminar series, Proteoform Thursdays, close to my heart. And yeah, I thought I would talk a bit about this concept that came up in a talk with Gloria here about proteoform systems biology, and hopefully we can develop new tools to kind of get at the roles of proteoforms in cellular subsystems and kind of highlight differences of function within proteoform families. I'll try to introduce what those concepts mean as well. And there's my Twitter. If you feel like mentioning me or contacting me, that's an easy way to do it. Proteoforms are the amino acid sequence of a protein with modifications at certain positions, and they're defined by those two things. Each gene can be expressed as a ensemble of different proteoforms, as you well know, and we can kind of call this a proteoform family. And on this slide, I kind of have shown kind of one way of depicting this, where genes are these pink rectangles. Alternatively spliced transcripts might be pink diamonds here, and they're translated into proteoforms or proteins, and then modified to become proteoforms. Shown as green circles, and those can kind of make a graph extending out from the gene. And so each of these individual proteoforms might have a slightly different function, and that's what we'd hope to investigate. And we have really good examples, as everyone knows here, of where proteoform variation can create different phenotypes or different functions. You have histones, you have histone clipping that change the accessibility of chromatin. Alternative splicing can make proteoforms from the same gene have as different of interactions as different genes completely. And phosphorylation is a key example of signaling modification. So these proteoform variations underlie all sorts of phenotypes, and that includes cell-to-cell variations that are that and subcellular variations that we'd hope to see. And so these are kind of some of the questions I've been interested in, how proteoform variation can change subcellular localization, how it can change, the expression of proteins, between single cells, and also, how it can create variability between different, between protein expression within tissue context. And so, yeah, today I was kind of hoping about talking about a tool I built for with a great team at Lloyd's lab for finding variant proteoforms that have amino acid sequence variations using this tool Spritz. And then I'll talk a little bit about applying that to the blood proteoform atlas dataset and integrating the human protein atlas, human blood atlas dataset to try to find proteoforms that are the result of alternative splicing. And then I'll talk a bit about imaging proteomic data, kind of a different data modality from top-down proteomics. And hopefully we can use some of this modality to look, into the cell and see subcellular features all the way down to, speckles and aggregates and, all sorts of interesting things that you can see within the cell. And yeah, and then I'll talk a little bit about this, potential for integrating imaging and mass spectrometry proteomics data. And hopefully in the future, we can use it to interrogate proteoform systems biology. So an overview of Spritz. This is a tool I built in Lloyd's group with the help of people there. And yeah, Spritz takes in RNA sequencing data and tries to produce a protein database that has amended sequence variations, and it's also capable of being it's capable, hopefully the database is capable hopefully of detecting peptides and proteoforms with sequence variations. And it's basically a pipeline. It's a snake make pipeline that automates the execution of a bunch of different tools to kind of at the end produce a database that can be used in search algorithms. So Spritz, implements a snake make pipeline. And I also customized a fork of this variant annotation tool called SnpF that has a lovely data structure for kind of representing transcripts and then using the RNA sequencing data to change those sequences and translate them into protein sequences. And then I also, this library is an internal library that's publicly available, open source, from Lloyd's group, and in here I updated or added some logic for really improving the way that variants are handled with appropriately including heterozygous variations or homozygous variations are appropriate, and also being able to modify the sites of variations. And so so I looked at some publicly available data sets for JERCAT. Deep RNA sequencing reads for 3 bioreps. I looked at a bottom-up data set and used this to kind of annotate sites of modification that we use for analyzing top-down results. And some interesting findings here is that some of the arginine variations are some of the most common ones, and so these are particularly important because they eliminate tryptic cleavage sites. So it's important to consider these if you want to expand the number of identifications that you see in bottom up, but also it kind of points to the value of top down, since hopefully you can just see these mass differences in the precursor mass of proteoforms. And we were able to see 43 variant proteoforms out of around 1,400 total. And this included one of these heterozygous variations where we saw an unmodified form and a phosphorylated form of SEC61B, both with and without this site of variation. And then it also includes a whole host of modifications in the results. Hopefully some of these are, I think, I don't know, take it with a grain of salt, some of the, combinations, but hopefully they're all, hopefully they're more accurate than not at 1% FTR. Yeah, and so I also applied this this Spritz pipeline to the blood proteoform atlas dataset in a collaboration with Neil Kelleher's lab to utilize the blood protein atlas that was produced as part of the human protein atlas, which I work with in Emma's group. And it's a pretty incredible data set. Usually I'm used to seeing 3,000 proteoforms in a data set, and this is almost 10x that. So it was very exciting to see. And one of the main questions was to ask, are these identifications comprehensive, or is there a lot missing from isoform variation? And so I integrated, an isoform discovery pipeline into Spritz and tried to use that to produce a database that was informed that contained, protein sequences from isoforms and see if we could see things that were missing. And one of the big findings here is that proteoforms are more specific indicators of cell differentiation than their corresponding proteins. And so if you look at these individual cell types, of blood cell types, you can see that there are a bunch of proteins that are kind of proteotypic for these different cell types, whereas for the proteoforms you have much more much higher specificity for each individual cell type. And so this really motivates the idea of looking at proteoforms for understanding cell type diversity. And I applied Spritz here to try to, again, figure out how many hits were missing in this data set, and produced a database that contained transcript isoforms, and merge those as merge those into, or translate those into protein sequences, and tried to find new proteoforms in this data set. And so we analyzed, the 12 blood cell types that were common between the human protein atlas's blood atlas as well as the blood proteoform atlas, and we actually identified slightly fewer proteoforms. So in this kind of subset of the data, we saw 4,600 or so proteoforms, whereas with the UniProt database, we saw 5,300, and we found 238 proteoforms that were identified only in this transcriptomic informed database that has these proteoforms that result from alternative splicing, which is only 5%. So we're not finding that many more with that database. And in this case, it was actually good. It's kind of a negative result that got me on the paper, I guess, because it kind of shows that it's pretty comprehensive. That just using the UniProt database is fairly comprehensive, and we're not missing a bunch. But I would like to emphasize that I think proteogenomics will become a lot more important as we go above the 30 kilodalton limit for denatured top-down proteomics that has basically been the cutoff for at least 5-10 years. I don't know. So we only find very few, like a handful of interesting findings below 40 kilodaltons, but as we go to larger proteins, there's going to be a lot more splicing variation. And I threw in these plots because I find them just like kind of stunning that there are genes that have 140 introns, and there's got to be a lot of splicing variation going on there. And these are very large proteins. So, as we go higher and higher on the, as we go higher and higher with the mass limit of top-down mass spectrometry, we'll really need to kind of interrogate this splicing variation. And I think that it'll become critical to really use this long read sequencing platform that Gloria is really demonstrating the utility of for isoform characterization. And this is a great paper by Rachel Miller and Gloria's lab. Yeah, that shows the power of this to kind of simplify the discovery of these isoforms. So I think that's kind of the way of the future to really get at this isoform variation in the proteoform landscape. And another kind of open question that I hope gets solved at some point is, this is a great paper that showed that there are fewer expressed genes by single cell RNA sequencing as cells differentiate. So as you get, as you go further and further along the developmental time for certain cells, as cells differentiate more, there are fewer genes expressed. But I think the real open question is: are the proteoforms, are the number of proteoforms stable, or do those go down? Do those get more specific, or are they expanding? And as you look at the proteoform families for each of these genes, are those expanding? Is there finer controls to each of these genes that are exhibited at the proteoform level? And so hopefully efforts like the Human Proteoform Project can answer these types of questions. Yeah, and I'd be fascinated to know the answer. Yeah, and so I'm in Emma's group, and we do imaging proteomics, and I think it's a really cool technology. So I think there's also questions we can answer hopefully with this type of imaging modalities. So I'll switch gears here to talk about some hopefully complementary methods to these kind of growing ensemble of top-down mass spectrometry methods, and hopefully we can observe proteomic changes that are informative of the of proteoform variation at the single cell and subcellular levels with microscopy techniques. And I'll specifically mention multi-localizing proteins and cell cycle proteins as examples of what I found and what I hope to find. And I'll talk about this potential of merging mass spectrometry data and imaging data into hierarchical models. Yeah, so I'll tie back to this a bit. That proteoform variation, I would take out this may, but I think it underlies single cell variations. It induces subcellular localization changes. There's translocations as the result of phosphorylations, and it changes single cell expression. There's and I think that a lot of the variability that we see in tissue context is largely the result of proteoform variation for each individual protein. And so the data that I'm working with is from the Human Protein Atlas, and this is a resource publicly available with antibodies against 13,000 proteins, which is more or less all of the non-secreted proteins. And it turns out more than half of these proteins are multi-localizing. They end up in many different compartments, and are contained within for example, cytosol nucleus, and there's a lot of complexity here to look into, and hopefully we can understand this variation at the proteoform level. And I think that part of the reason for this multi-localization is that these HPA antibodies represent or depict the superset of proteoform localizations. And HPA antibodies are designed to target as many isoforms from each individual gene as possible, to kind of capture where the protein is. This kind of larger concept of the protein, containing all the proteoforms. So as a result, the HPA antibodies are really targeting the superset of proteoforms, which I think is really cool, because it's not it's a nice design feature, because if you look at GapDH, I find this one image to just be so interesting, because it's quite well validated antibody, and GAPDH is just everywhere. It's in the cytosol, it's in the plasma membrane, it's in the nuclear membrane, it's in the vesicles. It's all over the cell. So in terms of housekeeping, if it's a housekeeping gene, then it's really, it's all over the house. Yeah, and so, I think that a lot of these variations, especially, nuclear to cytosol translocations and beyond, are the result of proteoform variation. And I think we should, try to develop methods to interrogate and select, proteoform families and look at and modification sites. Typo there, and hopefully we can develop technologies to interrogate, the effect of these sites on subcellular localizations. And pick apart, right? So it would be nice to know that one proteoform is with some certain modifications in the vesicles. It's really active and putting GAPDH into the vesicles. Some phosphorylation, for example, or that another is involved in the translocation. So one idea I have is hopefully at some point doing a targeted sort of prime editing scheme where we would be able to replace, for example, serine with glutamine, with glutamic acid, and be able to mimic phosphorylations and show the localizations of these two, and hopefully there'd be some significant differences that we could quantify and show as proteoform families, where one might be in the cytosol and one might be on the nuclear membrane or on the plasma membrane. Here I said nucleus for simplicity, but yeah. So this is that multi-localization problem. And I think this is something that'll be really interesting to interrogate in terms of proteoform variation. Another important type of variation is over the cell cycle. And it turns out that, genetically identical cells, so these are all U2OS cells, exhibit variability in protein expression. So if you look cell to cell, there's a lot of variability here. Some have very low expression of aniline in this example. One has it at the cytokinetic bridge, one has it expressed in the nucleus. And so, one big question in the Human Protein Atlas data set is, what's the extent of this variation at all? And another key question is that the cell cycle is involved here. And so how much of this variability is due to the cell cycle? And then how much is not cell cycle correlated? And that's a very interesting question as well. So to answer the question, the extent of these variations, around 1/5 of all proteins show the cell-to-cell variations. And these can happen at the level of expression between individual cells. And it can also happen in spatial redistribution, where it might move from the nuclear plasm to the nuclear speckles, for example. And these are robust. So around 70% of these variations are present in all the cell lines tested. And yeah, so with Diana Modesian, we did a kind of the first systematic study of this cell-to-cell variability and mapped the expression of these proteins to the cell cycle using some cell cycle marker fluorescence measurements. Yeah, and so we used two markers called this Fuji model to map each individual cell to the cell cycle. And so CDT1 is expressed and with our RFP in G1, and then in G2 we have geminin expressed, and at this S transition we get a yellow color. And so you can kind of see these cells progress from red to yellow to green as they go through the cell cycle. And in the data set, actually, it's not really a live image, but each individual, these are fixed cells. And so we're able to visualize and calculate the cell cycle position for each individual fixed cell. And one big advantage is that they're asynchronous, they weren't synchronized. And synchronization of the cell cycle actually induces a lot of cellular shifts And so this is kind of agnostic of these cellular shifts due to synchronization, which is good. And you can kind of see that the variability of these cell cycle markers is actually fairly reasonable over the cell cycle. So this is pretty accurate cell cycle time calculations for these many cells over the whole this whole U2OS Fuji data set. And then one other big part of this is that I integrated the single cell proteomic data and single cell RNA sequencing data to kind of get at these questions of whether or not these cell cycle dependent changes in proteins are tied to transcript variation, or if they're just dependent on the cell cycle. I'll get back to the proteoform stuff in a sec, but yeah, and then it turns out that we found about out of the 12, we found around 400 cycling RNAs. And a really interesting thing is when we calculate the cycling of RNAs and proteins, is that there's actually this 9-hour delay on average for each gene between the peak RNA expression and the peak protein expression. And so if you need another argument for studying proteins, it's instead of RNAs, it really reflects the or RNAs really don't quite reflect the full phenotype at an individual development time, since proteins it takes a while for the protein that does the work in the cell often to be expressed. And so so a lot of these RNAs are expressed in G1, and it's not until G2 that we actually see the protein at its peak expression level. And for proteomic variation, we found over 300 new cell cycle proteins. And kind of surprisingly, 72% of these were unexplained by the cell cycle. So, and I think this is a really interesting, very interesting thing to study. And, I think a lot of these cell-to-cell variations, some of them are going to be explained by proteoform level variation. And another really interesting finding is that only 15% of cycling proteins had, corresponding cycling transcripts. So a lot of this is really only observed at the protein level, and there's probably degradation going on, and probably PTM regulation, of signaling that type of degradation over the cell cycle, and then conversely also signaling the expression of the protein. And so this kind of forms an extended network of cell cycle proteins. You have this core network of transcriptionally regulated cell cycle proteins that are very, very well annotated, and then these new kind of non-transcriptionally these new non-transcriptionally regulated proteins that are that we found in this data set really start to form this extended network. They're less tight to the core, and so we're able to kind of expand this, what's known about the cell cycle and which proteins are involved. So an ongoing study here is that we isolated these 3 different phases and did a very deep phosphoproteomics analysis of this data. And we got a lot of, it's very deep, 77,000 quantified sites, which is pretty remarkable, in collaboration with Dan Itzak and when he was at the Chan Zuckerberg Biohub. And we saw over 2,000 genes with significantly changing phosphosites, and this forms once again, in purple. In yellow were those proteins that I showed before, and in purple are the cell cycle correlated phosphoproteins. And so once again, we get this kind of expansion of knowledge about the cell cycle with phosphoproteins. So yeah, I'm still digging into this, and hopefully this will kind of be fodder for future proteoform experiments, because this is bottom-up proteomics, and I find myself kind of struggling a little bit with, okay, just don't think about the proteoform level variation, how these are tied. It's just focus on the the site-specific information, because yeah, hopefully in the future we can, use these sites as candidates for, smart proteoform level studies. And finally, I think there's a big opportunity here with trying to reveal new cellular subsystems by integrating this mass spectrometry data and confocal microscopy data. And this is the result of a collaboration between the IDECR lab and Emma Lundberg's lab, the lab I'm in. I wasn't on the papers, but I think it's pretty fascinating. And so yeah, the cell, basically the idea behind it is the cell is comprised of a hierarchy of subsystems. You have proteins at the smallest level, and then complexes are collections of proteins, and pathways are collections of complexes, and organelles are even larger, and they have a bunch of several different pathways going on, and then the cell contains all that. And so basically embedding these physical protein distances based on protein-protein interactions and from confocal microscopy images, you can kind of capture this protein level and complex level interaction data with organellar and cell level data, and really reveal kind of new subsystems of genes. And so on this kind of multi-scale hierarchical map here that they built, these edges are kind of containment. So the cell contains the cytoplasm and the nucleus, and the cytoplasm contains these compartments. And in purple are these putative subsystems that were found. And so they were able, UHN and Yadi Collab were able to find new subsystems of proteins, and it's already moving forward. So this first one was with 600 proteins in HEC 293, and Leah Schaefer here is moving forward with this with a new and beautiful U2OS data set that has almost 10 times the number of proteins in it. So we're gonna hopefully learn a lot very soon about all the cellular subsystems of the cell, and hopefully find some new ones. And then hopefully in the future we can integrate proteoform level data with this, and maybe innovate some new ways for either integrating proteoform data or displaying proteoform level data to really pick apart, instead of having just individual genes here where you might see GAP-DH in the cytosol and plasma membrane and the nuclear membrane and vesicles. It's all over the place in the cell. We might be able to see that, oh, it really is just two proteoforms in the cytosol, and one is like transported in vesicles or something like that. We might be able to pick apart this multi-scale model more with this proteoform level data. So that's kind of a little vision. And so for acknowledgements, I have a lot of people to thank, and yeah, I worked with Lloyd Smith's group in my PhD, and had a blast looking at proteoform level data, and working with the great team there, and worked a lot with Michael Short-Reed on on doing some of the initial programming work, and Gloria Schenkman was a mentor for me there, and I really would like to thank her for all the discussions and that are ongoing, and just it's just a exciting field to be in with you. And I'd like to thank Rachel Miller. She helped me a lot with kind of debugging the Spritz code and making Spritz a functioning tool. And Leah, we worked together along a lot of, Leah's actually, we were in the Smith group together, and now she's in the IDECR lab, and we're working together on kind of a perspective Z review article, and there's just a lot of really exciting stuff happening. And then finally, of course, I don't know, with the blood proteoform atlas, thanks to the Neil Kelleher group, and that was a really fun collaboration. And and the Lundberg lab is very big, and it's been a lot of fun to work with Diana and Christian and Devin on the cell cycle work. And again, there's my Twitter. Feel free to reach out, and would love to chat with all of you. And Yeah, I'll be hopefully on the job market soon. So if you see any faculty or tenure track positions that look like they'd be a good fit, please, yeah, let me know. Be great to apply for them. Yeah, so thanks so much, and I'd love to hear your questions. Hopefully this sparks some ideas about proteoform systems biology and how we can kind of drive towards that.
Well, thank you very much, Anthony. That was great. I will leave it to you and the people in the chat how you want to handle their questions. People can either reread them, making the chat superfluous, or you can read them however you want to do it. Either way, I know that Gloria was first up, so you take the first question.
Sure. Great talk, Anthony. This is very exciting. So I have a couple questions about the imaging data. So a lot of proteins are multi-localizing. How many of those genes do you think the multi-localization is explained by different proteoforms?
Oh, that's a good question. Yeah, I don't think we know. But also we have like classic examples of where it does happen, where, there are genes that are phosphorylated at one specific site and then they're translocated from the nucleus to the cytoplasm. And so just based on, how much of these changes we already know in the literature that are, from one specific phosphorylation, I think it's actually probably the majority. That'd be my guess.
Yeah. Because I think it's a really interesting, it's a fundamental question. To what extent does protein localization, to what extent is it driven by molecular variation versus interactions? You can have the same protein, but different environments.
Definitely.
As local protein interactions or other molecular interactions.
Yeah, and
the localization.
And also how many of them are, are both, where the interactions are changing because of modifications and yeah, I think also, the structure of the protein can probably change as a result of interactions and modifications, and that kind of can drive, changes in interactions and get, translocation events as well.
Thank you.
For example, like disorder regions folding up because of interactions, and so definitely both can play a part. Yeah.
Thank you.
Yeah, thank you. Should we go to the chat?
Yeah, exactly.
Awesome. Oh, okay. Gloria, questions on this slide. What were the proteins only in UniProt? And what properties define only those found in UniProt and not the proteogenomics approach? So this is probably the blood cell atlas. And yeah, I think I'm not sure I have the answer to this, but we did find, some one or it was really just a handful of interesting kind of findings with the proteogenomic data in this case. We found one really compelling example of kind of a ribosome protein that had a little splice junction in it that we found that seemed to be an interesting finding. So there's one little transcript variation that was interesting. But yeah, it's kind of the conclusion of that was just really that we didn't find much, and that the blood proteoform atlas is quite comprehensive. And so it's really kind of looking forward to the future of seeing these larger proteoforms and all the splicing variation with the
Yeah, the motivation for that question is that you might shed light on what needs to be worked on, because there were a couple hundred proteins that were only found with the UniProt database. So that means there's something that's not the signal that's not coming through from the transcriptomic side. So maybe that could be inspiration for like new projects.
Yeah, with the I actually didn't merge in all of the UniProt sequences. I just used the proteogenomic database from the transcript model, and the transcript model had the reference sequences, so that this was based on ensemble, and so UniProt has this kind of like lovely curated set of protein sequences, and ensemble is a little more chaotic. So I think it's kind of some of those differences are just due to the fact that it's kind of based on a different reference.
Got it. Thank you for clarification.
If I could ask a question?
Yeah.
I don't know if you can hear me.
Yep, I can hear you.
Okay, great. Great talk. I was
Thanks.
glad to be able to join it. Had to fumble through my get a different computer because I couldn't get the Zoom to operate. But I'm glad I'm here. You showed this this slide with regard to the temporal difference between the RNA and the protein production. Mm-hmm. That to me was pretty amazing to see, because it kind of begs the question of maybe including some information or informatics to be able to have like a temporal protein database when searching, based off of RNA signal, or having a time component in the database would be helpful when analyzing laser microdissection samples or something like that, because it was pretty astonishing to see the shift.
Definitely, yeah. I mean, yeah, you mentioned laser microdissection. Yeah, I think it's critical because we have all this kind of like growing up, bundle of spatial transcriptomic techniques, and when you look at the tissue level, you're not really, with spatial transcriptomic techniques, you're not necessarily going to see the protein. It might take a while for that protein to be expressed, and
right.
Yeah, it's a great thought.
The other question I had was with regard to the multi-localization. I wonder for some of these reagents that are being used, do you know whether or not there's cross-reactivity playing a role in that, where for certain antibodies there may be, there's the intended target, but with sequence similarity you may be lighting up different areas based on a conserved region. I'm just curious to know your thoughts on that.
I think they if you look on I should have put these plots on, but I don't quite understand them quite well enough to put in a talk. But if you look on the Human Protein Atlas website, for each antibody, I'll try to pull it up while I'm saying it, but for each antibody, you have this list of domains, and I think they're kind of constructed, or the antigens are designed to be. All right, I have it. They're designed to kind of target the regions that are unique to the protein, mm-hmm. But also sort of covering domains that cover different transcript isoforms as well. So this one antibody kind of covers these these domains that are kind of unique to this protein, but this is why I say that I don't quite understand it well enough to put it on top, because I'm still wrapping my head around all these.
No, but it's a great it's a great point.
Yeah,
especially when you're seeing these beautiful images, to be able to understand how what is leading to that signal, and what could be sources of additional sort of background in the sense it's still good information, right?
Yeah.
Especially if you know that particular antibody not only binds this protein, but it also binds a different protein. And so seeing that image in the context of understanding what that antibody interacts with could be helpful, because you may be able to sort of tease apart. Cross-reactivity no longer is an issue there, right? Because you may know that later on that second protein, that cross-reactive protein, is present at a different phase. So it lights up a different region of the cell, which correlates to the localization of that cross-reactive protein or something to that effect. So I think that additional information is very useful. It could potentially allow you to tease more away from more information away from the images that you're seeing by no longer treating the cross-reactivity as a dirty word, but just as more information.
Can I jump in here? This is a really good, within our lab, we're trying to, I just realized what you're talking about is a patch on the protein. You call it an antigen, but it's a patch on the protein. And when you talk about comparing proteoforms, we actually are looking for a word for this. When you compare two proteoforms, ultimately there's, different sources of variation, phosphorylation, splicing, but we're thinking that what matters is that bit, like what the cell sees, that bit of sequence that's altered. So when you say cross-reactivity, it's actually, this is all one and the same with the biology that we're interested in.
Yeah. Yeah, and the thing is that the sequences don't need to necessarily be the same. The you could have different electron sequence divergence, but
very similar shape,
similar electron donors, electron acceptors, where you have that single, an arginine for a lysine, or maybe a histidine is placed there, but you still have that donor there that is able to interact with the antibody, because so the sequence alone may not tell you the cross-reactivity. And so that's why I'm sort of getting to, maybe for these types of reagents, it would be helpful, I think, to have some type of IPMS characterization of the reagent, so that you can say, with this reagent, I not only pull down the target protein, but I also see in a number of different cell lines with positive and negative controls, these other two proteins, and it's pulling down, it's interacting through this peptide or this sequence or this region. And so now I know that reagent gives me a handle or a readout for these 2 or 3 different proteins. Now, it might be weaker in terms of binding strength, but it's still going to light up, especially if that other protein, the primary protein, is not present.
Yeah.
The community here. So we need a word for this, because antigen doesn't cut it. Patch, proteoform patch. We need a word to talk about that bit of surface, the cell that's biologically relevant that the cell sees, because that's actually a subunit of proteoform, if you think about it. And Dr. David Cooper here is a research assistant professor. My group is coming from the structural proteomics background. And so, it's kind of related, there's this imaging part, but then the structural biology part. And I don't think David, there's a word for this, right? You talk about domains, structural domains, intrinsically disordered domains, interfaces, patch.
Someone said epitope, which is pretty funny because
proteoform epitope. I guess it is epitope, but then you're defining that question in terms of what antibodies can recognize. What we're talking about is when there's 2 proteoforms, the difference could be really small or it could be really large. Whatever that thing is, it can't be bounded by what an antibody can recognize. Just spitballing here. We need a word.
Yeah.
If you have an idea, please tell us so we can start using it in our papers.
Frederick, if he's awake, no, it's much later where he is. I know he's awake, but he always has good ideas on verbiage like this.
Yeah, I'll give this one a think. Although epitope isn't that bad, I think.
I mean, I think there's a larger concept in it.
I understand what you're saying in the sense that epitope, you want something that sort of highlights the fact that there's multiple variations, or it's not just one static sequence, or it's more than one. It's a
proteotope.
Proteotope. Oh my gosh, I like that.
Proteotope? Did we just invent a new word? David, I think we're gonna use proteotope.
That's pretty good. Okay. Well,
Yeah.
any other questions before we go? That's
Yeah. Gloria, yeah, definitely nuclear localization signals play a role. Cross-reactivity, yeah, great question, great discussion. Can we elaborate more about the time lapse? Yeah, we discussed that too. Yeah, thanks so much. Yeah. IT seems like Mike isn't working. But yeah, I really like Proteotope too. It's just so fun.
yeah.
And yeah, it's such a great thought about the electron, the electron donor similarity and all these other, even modifications, right? Like if you put an acetylation on, you might induce the proteotope to be similar to some other protein, and yeah, or some other isoform. Great.
All right. Well, hey, if you're going to start using the term, you might as well write a paper about it and throw everybody on, because the proteoform is really.
Yeah, I'm gonna do a screenshot. Y'all are co-authors.
Improved everyone's H index. Everybody that was on the proteoform paper had their H index went up by 1, no matter how much else they published. So just a little word of advice. Okay. Any other questions? If not, well, first let me just ask the question. No. Well, great audience, great questions. And Anthony, thank you. That was amazing. And you have some applause, and it's so great to see all the people, old friends in the audience, and new friends. So with that, let's conclude this Proteoform Thursday, Proteotope Thursday,
Friday.
inaugural Proteotope Thursday. All right.